Back in episode 2, we flagged biologics as a parallel track to small molecules, and named a few players in passing: ESM3, Chai, Generate Biomedicines, ABSci. This episode gives that track its own hour, because biologics are a large and fast-growing share of new medicines, and the AI methods behind them differ in real ways from small molecule design. So start with the distinction. Small molecules, the subject of episodes 8 and 11, are tiny synthetic chemicals, usually taken as a pill, that fit into a binding pocket. Their design problem is navigating chemical space. Biologics are something else entirely: large molecules produced by living systems, like antibodies, engineered proteins, and peptides. They're far bigger, exquisitely specific, and their design problem is protein design, choosing an amino acid sequence that folds into a shape which binds the target. That's a different search space and a different class of model altogether. Antibodies are the marquee case: Y-shaped proteins whose tips can be tuned to grip almost any target with high specificity, which is exactly why they dominate modern biologic medicines. The headline generative model here is ESM3, from evolutionary scale. Its distinctive property is being all-to-all across sequence, structure, and function at the same time. Think back to episode 7, AlphaFold read structure from a sequence. ESM3 goes the other way: it can generate a brand new sequence to hit a desired structure and a desired function. AlphaFold read structure, ESM3 writes it. And the proof point is striking: a novel fluorescent protein they call ESMGFP, one that does not exist anywhere in nature. That's concrete evidence these models can generate genuinely new functional biology, not just remix proteins that already exist. The commercial signal is loudest right here. Chai Discovery's platform, Chai-2 and Chai-3, partnered with Eli Lilly in January 2026, and was separately licensed to Pfizer for next-generation antibody design. Generate Biomedicines is pursuing a generative approach to protein and antibody therapeutics. And ABSci, which has been publicly traded since 2021, has its AI-designed antibody, ABS-101, already dosing patients in Phase 1, one of the more advanced clinical data points on the biologics side specifically. Notice the same pattern we saw in episode 4, Lilly and Pfizer both buying into AI antibody design, spreading their bets rather than backing a single winner. Why is this such an attractive place to point AI? 2 reasons. First, specificity. Antibodies can hit targets that small molecules simply can't grip well, including the large, flat protein-to-protein interfaces that a tiny pill just slides right off of. Second, they've historically been slow and painful to design. Traditional antibody discovery meant immunizing animals or screening enormous physical libraries. Generative design promises to propose high-affinity binders computationally, and then validate just a small set at the bench, the same predict-then-validate throughput story that runs through this whole series. And the caveat from episode 5 applies here too: sometimes harder. Biologics are more expensive to synthesize and to assay than small molecules, so the wet lab bottleneck is even tighter, and the clinical data, ABS-101 in Phase 1, is still early. The flurry of big pharma partnerships is a genuine confidence signal, but it is not efficacy proof. Keep those two things separate, exactly as we did in episode 4. The money tells you smart people are betting, the trials tell you whether they were right.